Okay, great. So I think others are a bit late to join. But anyways, let's let's begin. So Alia has joined in. So do you have any doubts, Aisha?
I was not there in the last class, so I need to watch the recording.
Sorry, come again. Your voice is a little muffled.
So I was not there in the last class, so I need to watch the
You
recording.
okay? You're not there. So please please watch the recording and get back to me with any doubts if you have. Okay. Good evening, Alia. Okay, so let me do the recap or summary today, since Aisha was not there in the last class. So, okay, so in the last class, we talked about the replication machinery and how it works. So we know that DNA replicates, and for the DNA to replicate, the enzyme on which it depends for replication is called DNA dependent DNA polymerase, right? That's the enzyme that's responsible. And most important enzyme is polymerase. Apart from that, it's not just polymerase which can single-handedly replicate the DNA. There are lots of enzymes that do other functions. For example, you have to open this double helical structure, which contains two backbones, two chains in the DNA bound together by hydrogen bonding. So in unwounding, the topoisomerase and the helicase helps, it opens. So these two enzymes help in opening up the double helical DNA and make it a fork. So this, what you see here, is known as replication fork. So if in exam you see a question that draw a label diagram explaining replication fork, this is what you have to draw. Okay, you don't have to draw this neat and sophisticated. You can also draw it as simple as you can. But remember to include certain things, especially helicase, DNA polymerase. After helicase opens, I told you that there are these single strand binding proteins that bind to these opened single chains and stabilize them, right? And other enzymes that play important role is DNA ligase that joins together the Okazaki fragments. I told you about DNA polymerase that it has a directionality in which it works. So DNA dependent DNA polymerase works in 5' to 3' direction, right? Which means it can polymerize a DNA in 5' to 3'. Is that clear, everyone?
Yes, sir.
Yeah. Now, because of this limitation, what happens is that when the two chains open up, the two chains of the two, what should I say? Yeah, the two chains of the DNA have different polarity. One chain is from 5' to 3', the other is from 3' to 5'. And because of which, on one strand, on one chain, the DNA synthesis can be continuous, which is here, and it is called the leading strand. Because there is no issue, the polarity of the parent strand is 3' to 5', and therefore polymerase can work in 5' to 3' direction and keep polymerizing DNA one after the other nucleotide. Okay. But on the other strand, there is a problem. There's a little bit of an issue because when the double strand opens, the polarity of the parent strand here is 5' to 3'. And it is the same polarity in which DNA polymerase also works. Now, here the DNA pol has to wait for opening of the fork. So when it opens a little, it polymerizes in this direction, 3' to 5'. And then again, the fork opens a little bit more, and then it again makes a short stretch of strand and continuously keep making small strands, right? Now these strands are fragmented, so it's not a continuous run. And therefore you need DNA ligase. Is that clear, everyone? Am I audible, Aisha and Alia?
Yes, sir.
Okay. Now, in today's class, we will be talking about the second step of information flow, genetic information flow. So after replication, the next thing is that from DNA, also cells make RNA, right? That is called transcription. Now, in this case, the enzyme which is responsible is called DNA-dependent RNA polymerase. And I hope it is clear why, right? Because the enzyme is dependent on DNA to make an RNA, right? But it does similar thing. And this also has a directionality. So this enzyme also works in 5' to 3' direction. Okay, so here we discussed in the last class that when a DNA strand opens, so Aisha was not there in the last class, but Alia, you were there in the last class, right?
No, sir.
Yes or no?
No, sir.
Oh, you were also not there. Okay. So in the last class, it's okay. So in the last class, I asked a question. And let me repeat the question for you as well. The question was, see, in case of replication, we can understand that the directionality of the enzyme can cause a problem. But let's say in this case, this is a DNA helix. And let's say this is the sequence A, G, T, A, C, C, T, A, C, C, G, T, C, G. Okay. So this is the sequence on one strand. Complementary will be the sequence on other strand. You can figure out. So can you figure out what will be the sequence on this strand? If this is the sequence on, let's say, this strand, which has the polarity 3' to 5', this is the strand which has the polarity 5' to 3'. So what will be the sequence on 5' to 3' strand? Good evening, Iram. Glad you joined. Am I audible to you, Iram?
Yes.
Okay, great. Good evening. So, and also the spotlight feature is on for you. The question is, what is the sequence on 5' to 3' strand? Anyone? Someone wrote in the chat, and that's correct sequence. Yeah. So Alia has written in the chat. Alia, that's correct. That's the correct sequence. But the question here is, with which we ended the last class, and Iram was there. You all can write down the question. In transcription, we figured out and we know that only one strand of the DNA That also is what the definition says. It is the process of copying genetic information from one strand of the DNA into RNA. Now the question is why just once? Why from one strand? So write down why only one strand is copied during transcription. Why only one strand of DNA? Or why not both strands? The question can come in any manner. Why only one strand of DNA is copied? And during transcription. Anyone? So I think Iram is there and Mariam has also joined in. So Mariam and Iram were there in the last class. Did you figure out the answer for this? Did you think about it, Iram and Mariam? Anyone? Yes, no, say something. Iram and Mariam, are you there? Can you hear me, Mariam? Yes. Could you go back and think about this question that we left with in the last class? That why not both the strands copied during transcription? So there are two strands. One will have this sequence, the other will have the complementary sequence. So why not both the strands are copied during transcription? Why just one? Because you can say that because in the last class we talked about this enzyme having one direction, so that you can say that this strand, which is 3' to 5', will be chosen and it will copy information in 5' to 3' manner. But this problem does not holds its ground here, because once the DNA strand is open, both the strands can be copied in 5' to 3' fashion. Here we don't have to replicate the whole DNA or copy the whole DNA. We just have to copy a segment of it. So that's not a problem. But still, only one strand is copied. Why? So there are two reasons for it, two biological reasons. First reason is, if both the strands are copied, then the mRNA which will be formed will have different sequence. Okay. So from the same part of the DNA, let's say from this part to this part, it is the sequence of gene X, let's say. Now it has two strands. So if you make RNA from both the strands, there'll be two different sequences of RNA formed, right? And when those two different sequence of RNAs will be read by ribosomes in the cytoplasm, it will also produce two different kinds of proteins, which cannot happen and should not happen, which is, it's a problem. Okay. If you make two different kind of proteins from the same gene, then it will complicate the genetic information machinery. Okay. So for same gene, there has to be only one protein, one type of protein in the cell, right? Does that make sense, everyone? Do you understand? Yeah. So write down, the first is, if both strands are copied, then they will code for two RNA molecules with different sequence. two RNA molecules with different sequence. Okay, because why it happens? Because the two strands of DNA are complementary. They are not identical. So complementarity does not mean that they are identical. Remember this, they are complementary to each other. They do not carry the same sequence. Okay, they carry opposite sequence. So therefore, the transcription machinery has evolved in a manner that molecularly it can only read one sequence and not the other one. Okay. And what is the consequence of this? Write down. Two different RNA sequences. Two different RNA sequences coming from the same segment of DNA. Two different RNA sequences coming from the same segment of DNA will further code for two different proteins. Will further code for two different proteins. Does that make sense? Makes sense, right, everyone? No one is speaking today.
Yes.
Yep. Okay. Now you can add one line that this will complicate the the flow of genetic information. This will complicate the flow of genetic information. Okay. And what is the second reason? I told you that there are two reasons. So one reason is that two different proteins, you will end up forming two different proteins. That's why in the cell, two RNA should not form from the same segment of the DNA. Now, this is the application-based problem in biology. If this happens, then biology will not function normally. But the other reason that it also cannot happen is, see, RNA is single-stranded or double-stranded? It's a single-stranded molecule, right? And if there are two single-stranded molecules having complementary sequence, so basically the RNA that you will make from this strand marked in green will be same as the sequence of this strand, which is marked in the red, correct? And the RNA which will be formed from the red marked strand will be same as the sequence of the green. So at the same time, there will be two single strands, right, produced in the cell from the same segment of gene. And these two single strands will be complementary. This is the problem, right? What will happen? What will happen if two complementary single strands? Yes, Alia says a dsRNA. It means double-stranded RNA. So RNA is single-stranded, and if it finds its complementary pair, it will go and bind with that to become stable. And that's a problem. If it becomes a double-stranded RNA, then it can't be read by the ribosomes. And this whole mechanism in which a lot of energy is used to copy the information will be a waste of energy. So at the end, you will fail to make any protein. So this is the second reason that you can the transcription machinery should not and cannot read both the strands of the DNA simultaneously. Okay. So write down the second reason. The two, if both strands are copied, then two complementary RNA molecules will be produced simultaneously. And because they are complementary, so they will form a double-stranded RNA. They will form a double-stranded RNA. Okay. And you can write down a double-stranded RNA can't be translated into protein by the ribosomes. Is that clear, everyone?
Yes.
Okay, great. So anyone has any doubt?
So,
Yes, Iram.
so you said that the sequence of RNA at 3' to 5' end would be similar to that of the sequence of RNA from 5' to 3', right?
No, I said the RNA that will be formed from the 5' to 3' DNA strand will be, the sequence will be same as the 3' to 5' DNA strand. So the RNA formed from this strand, which is marked in red here, will have same sequence as this strand, no? Because you are making a complementary RNA, and this is already a complementary thing. You understand?
Yes, sir, but still uracil will be there instead of thymine, right?
Exactly. So uracil and thymine are very, very similar structures. The only difference in uracil and thymine is of a CH3 group. If you go and look at the structure of uracil and thymine, you will see that they are almost same. And that's why this is one of the proofs in evolution that thymine evolved later from uracil itself. Okay. So in RNA,
Yes.
there is no concept of thymine. It's always uracil. So even here, when you read A, when the transcription machinery reads A, it cannot add a T in the RNA. It can only add a U in the RNA. Okay. Does it make sense?
Yes, sir.
So do you understand that the complementary sequence will be like this? The complementary sequence of this green marked strand will be similar to the other strand, and the complementary sequence of this red marked strand will be similar to the other strand. The RNAs will actually be similar to opposite strands itself. Hence, they will also be complementary if both of the strands are read together and transcribed. So they will join together and make a double-stranded RNA. And this whole process will be a futile process. And cell cannot survive if you have futile processes using too much of energy. Okay, make sense?
Yes, sir.
Okay, that's good. So let's move forward. So now, since you know this cannot happen, and we know that only one strand is read, let's go and learn more about the process and this strand. So I'm going to teach you about the transcription unit. Just like we had the replication unit, the machinery, We're going to study the transcription unit. Now, in transcription unit, what you have to understand is there are three regions. Wherever in the DNA, you need to start transcription. Mostly these are genes that are transcribed into mRNAs, and then mRNA form the protein. So we say that the gene is expressed. To give you an example, so you all have the gene of melanin in your skin cells, but when you're exposed to sunlight for very long hours, the presence of sunlight, the intensity of sunlight stimulates your skin cells as a physical factor, and the gene of melanin gets transcribed more and mRNA is formed, and that mRNA is translated into protein, and you make lots of melanin in your skin cells, and hence we get tanned. Okay, that's to protect us from high intensity sunlight. You all must have experienced it at some point or the other, right?
Yes.
Tanning, yeah. Then again, when you come back in shade and you start living a normal life, no more exposure to sun, in some time the tanning goes away, right? You don't have to worry about tanning, because it will go away if it's not your natural skin color. So people have inherent by birth or natural skin tone. Even the skin tone with which you are born can lighten up or darken up depending on in which region you are living. Okay, but your skin tone mostly operates in one particular tone, and it can go up and down based on where are you staying, which area of the world you are staying in for a longer time. But that deviation will always be coming back to your normal skin tone. It's easier for a light tone, fair skinned person to tan. Comparatively, it will be difficult for a dark skinned person to lose all the melanin. And because I told you that the skin color is a polygenic inheritance, right? We talked about this in last chapter. So if you have multiple genes and multiple alleles that are dominant, then you will have a darker skin shade. Okay, so let's come back to transcription unit. So write down any region of the DNA when we define as transcription unit, it should have the three regions. Okay, so it should have three things. One is promoter. So there should be a promoter sequence, and this is where the initiation of transcription begins. Then the second thing it should have is a structural gene. Basically the gene that you want to transcribe, why you are doing all this business. So for this particular thing. And then once transcription is done, there should be a terminator region. Okay, so these three regions should be there: promoter, a structural gene, and a terminator. Okay, and if I draw it for you, then let's say this is a double-stranded DNA. Okay, it's a double-stranded DNA. Let's mark this as 3' to 5'. It tells you that in this strand, phosphates are on your left-hand side of the screen, and OH group is on the right. Here it's 5' to 3', which means it is opposite. Now, the strand which is 3' to 5' is called the template strand. Okay. Why it is called the template strand? Anyone can guess? What does the word template means? Quickly. Template means which provides a base for like any enzyme to sit there and work. Like in case of replication, both the strands, the parental strands were called template strands, right? and then new strands were synthesized. Here only one strand is called template strand and it is the 3' to 5' direction one, which is called template strand. Okay, because on this strand, the RNA polymerase can sit here and start transcribing information in a 5' to 3' manner. You understand? So this will be the new RNA forming. Does it make sense, everyone? Okay. So template strand act as a template for DNA dependent RNA polymerase. And towards the 3' end of the template strand, there will be a region, the sequence, which is present here. This will be called promoter region. And promoter region is the region where RNA polymerase comes and binds. Okay. So here the RNA pol comes and sits. So this is DNA dependent RNA polymerase. And then it's from here, the transcription starts in this direction. So this is called transcription start site. Transcription start site. And it will go all the way till the end. And at the end, there will be another region towards the 5' end of the template strand. And the sequence which will be present here will tell the RNA pol to now shed off. Okay, get off the DNA and go away. So this is called the terminator region. And this is where transcription stops. And everything in between of this is called the structural gene. Structural gene. So this is the gene which we are transcribing. That's why we are doing all this business. Okay. Now the other strand, the opposite strand, is called coding strand. Now this is a don't get confused by the name, because this strand does not code. It is not read by the transcription machinery, RNA polymerase. But can you guess why it is called coding strand then? People? Why the strand 5' to 3' is called coding strand? Because the new RNA, which will be transcribed from the template strand, the sequence of that, the code present in that will be same as the coding strand, except at every place where there is a T, the RNA will have a U, but the sequence, the code will be same. So coding strand is called coding strand because it codes the same sequence that the mRNA will have. Make sense?
Yes.
Yes. Alia says maybe because 3' to 5' will code it. 3' to 5' will code for a mRNA, and the sequence of that mRNA will be same as the sequence of the coding strand. You're very near the right answer. Okay. One strand of DNA cannot code for the other strand of DNA. Does it make sense, Alia? Do you understand what I'm saying?
Yes, sir. I was just trying to.
You're trying to say the same thing. I know.
Yes, sir. Yes, sir.
We're not finding the right words. I understood.
Yes, sir.
But remember, if you write things in boards, write it clearly, because Rahul will not be checking your copies so that he can understand what you're trying to say, because you're his student. So the one who will be checking your copy will solely rely on your words. So clarity of words is very important. Sometimes we know the right answer, but we don't get the deserving marks because we're not able to express it in right words. See, we are just like biology. If RNA polymerase does not do the work very correctly, and when it is transcribing, it makes some mistakes, will you be making the same protein? No, right? If it fails to express exactly what it should be expressing, the protein will change, the phenotype will change. Instead of having, black eyes, you will end up having blue eyes. Similarly, if you don't write the answer correctly, if you don't express your answer in correct terms, then instead of getting 5, you'll get just 3 or 2. Does that make sense, Alia? Yeah. So this is a schematic of a transcription unit. So promoter, structural gene, and terminator region is there. This is how we show the direction of transcription by an arrow, which goes from the promoter region towards the terminator region, and promoter region is where the RNA pol sits. Okay. Okay. So coding strand, you can just write also coding strand. It doesn't code for anything. And template strand, you can write for template strands. It codes for the mRNA. Okay. Perfect. Now, on the basis of this transcription unit, we also have a definition of gene, a different definition of gene. So let's pay some attention to understand what do we mean by gene. What is a gene, everyone? As you can see here, I told you that the region between the promoter and terminator is called a gene, right? Structural gene, we call it. Is it clear, everyone? So you can define gene as a, can I say that gene is a functional unit of inheritance? Can I say this?
Yes.
Yeah, right. So, but we know for sure that where is gene located, right? You can actually find promoter regions in our own genome, and you can say that this is where a gene starts and this is where a gene ends. And we call that as open reading frame, right? I told you about this, that every gene needs to have a start codon, which is AUG, and a stop codon. And that's where the open reading frame is, and that's called the gene. But in terms of DNA sequence, it is very difficult to define a gene, right? So another thing is, let me put it like this. So we know that in transcription from DNA, we make RNA, right? But how many different types of RNA do you know about? Do you know about just one RNA? Have you heard of tRNA, rRNA, mRNA, these terms? Yes or no?
Yes.
Okay. So what is a mRNA?
Messenger.
Messenger RNA. And why do we call it a messenger RNA? Come on, tell me. See, words are very simple. If you're not going to talk, then it's going to be a boring class. I'm just talking and you're just listening. Why do you think it's called a messenger RNA?
I think because it carries a message from nucleus to cytoplasm. I think.
Exactly. Perfect. So if you knew the answer, you should have spoken earlier, right? So speak as you get the opportunity. You're right. Messenger RNA carries a message from nucleus to the cytoplasm. And what happens in cytoplasm? Who reads the message? Ribosomes catch the mRNA, read the message, and convert that message into a polypeptide chain called protein. Okay. And that part is called translation, which we will be studying the next. Okay. So messenger RNA is the RNA that has the capability to carry this message, go to ribosome, and this message gets read by the ribosome and a protein chain is produced. What is tRNA? Where have you heard tRNA? Transfer RNA. And why is it called transfer RNA? Because it transfers something. Now, what does it transfers? It transfers amino acid to the ribosome so that it can make a polypeptide chain. But remember, tRNA itself is not read as a message by the ribosome. So tRNA helps in protein synthesis, but this is not the RNA that codes for a protein. Okay. The last is rRNA. There are others also, but just for the sake of simplicity, what is a rRNA?
Ribosomal.
Ribosomal RNA. Where is this present and what does this do?
It is a part of ribosome.
Yes, it is a structural part of ribosome itself. So ribosome is part RNA, part protein. It is an organelle which is made up of RNA and protein only. It does not have a membrane around itself. That's why it's called non-membranous organelle. And hence it is present both in prokaryotes and eukaryotes. So ribosomal RNA is the structural part of ribosome. So it again is not the RNA that codes for protein. But this also helps in protein synthesis. Process cell called long non-coding RNAs or small RNAs or silencing RNAs. We'll talk about it later. But of these three, only mRNA is the RNA that codes for protein, right? Or proteins directly. Is it clear? Others do not code for proteins. But all these three kind of RNAs are produced or manufactured in the same manner through transcription. Does that make sense, everyone? So from a structural gene only comes a mRNA, comes a tRNA, and comes a rRNA, which means all the three are formed from structural genes, but all type of genes do not code for proteins. Is it clear to you?
Yes, sir.
Yeah. So now there is a problem. We used to think that all gene codes for protein, but that's not true. So that's where we need to now define the segment of the DNA or gene that specifically codes for a protein Rest other, we can continue, we can still call them structural genes, but they will not code for protein. They will code for tRNA or rRNA. Yeah, or other forms of RNA that cannot be read by ribosomes. So now came the concept of cistron. Anyone know about cistrons? Cistron is a word that doesn't sound biological to me. It's like electron, proton, cistron, neutron, right? Sounds very physical. So what is cistron? So cistron is the segment of DNA that codes for a polypeptide. So the gene that specifically codes for polypeptide or mRNA is called a cistron. So write down. Cistron is defined as a segment of DNA that codes for a polypeptide. Polypeptide is simply, it means protein. Just like polynucleotide is DNA or RNA. It's polypeptide, which means if you are transcribing a cistron, then you will make mRNA. And cistron is also a type of structural gene, but not all genes are cistron. And if you are transcribing something which is not a cistron, then you will not make a mRNA, you will make a tRNA or a rRNA. Is that clear? People, is that clear?
Yes.
Yes, sir.
Yeah. Now, a cistron, there are two types of cistronic systems in biology. First is monocistronic organisms. Now, monocistronic, which is mostly eukaryotes, including humans. Okay, we are monocistronic. Now, what does the word mono means? Let's go to the word. Mono means, yes, single or one, and cistronic means cistron means, it's written just above it in green, a segment of DNA that codes for a protein. So in humans, the way our transcription units are organized is that we have a promoter region, we have a terminator region, and all our structural genes are monocistronic, which means between one promoter and one terminator, we will only have one gene that will code for one protein. Is it okay?
Yes.
Yeah, so write down. Monocistronic structural gene, monocistronic structural genes code for a single polypeptide. Monocistronic structural genes code for a single polypeptide. So it's present in eukaryotes. Now, it has one more, it is an evolved system, so it has one more complexity. Monocistronic structural genes have interrupted, right? Interrupted coding sequence. Okay, now this is very, very unique feature of eukaryotes like humans and all other eukaryotes. So all our genes have interrupted sequences. And what do I mean by interrupted sequence? It means that if this is our structural gene, okay, before this is the promoter region, here is the terminator region. In this structural gene, you will find regions in between which are not required to make protein. So these sequences which are not required are called introns. They interrupt. And the sequences which are required are called exons. So exons and introns. So all our structural genes, basically what happens when transcription happens in humans is our structural genes are small. Like it gets shorter because all the exons 1, 2, 3, 4. 1, 2, 3, 4. So let's say this is exon 1. This is exon 2. This is exon 3. And this is exon 4. Okay. And this is intron 1, intron 2, and intron 3. So this structural gene, let's say in a eukaryote, has 3 introns and 4 exons. But after transcription, okay, post transcription, all the exons, exon 1, exon 2, exon 3, exon 4, will be stitched together, and all the introns will be removed. It will be cut and removed. So this is intron 1, intron 2 and intron 3. So all these are removed. And then this is our processed mRNA. This forms our mRNA. We don't need introns in our processed mRNA, which can be read by the ribosomes to make protein. So this kind of arrangement is called interrupted sequence. Okay, so write down. The expressed sequence are called exons. The expressed sequences are called exons. And they appear in the mature and processed mRNA. And they appear, and they always appear in the same sequence series. Okay, so E1, E2, E3, E4. In between, you will cut down introns and remove it. And next line, write down, the exons are interrupted by intervening sequences called introns. Intervening sequences called introns. That do not appear or which is not present in a mature and processed RNA, which is not present in a mature or processed RNA. So this is called split gene arrangement. Split gene arrangement. Is it clear, everyone? And it's present in eukaryotes like us. It's more complex. And this complicates the definition of a gene with respect to the sequence. Okay. Make sense, everyone? Okay. Now the second type of cistron is called polycistronic arrangement. A polycistronic structural gene. And this is present in prokaryotes, like a bacteria. Now, in the polycistronic structural gene, what happens is there is a promoter and there is a terminator. And in between, what does the word poly means?
Many.
Yes, many or multiple, right? So in this case, what happens is you have a structural gene. Before that, you have a promoter. At the end, you have a terminator. And in between, this is the structural gene, right? But in that structural gene, from this point to this, there is one gene. From this to this, there is one gene. And this could be one. So there could be gene 1, gene 2 back to back, like a cassette. Remember earlier there were cassette reels in which there are songs coded one after the other in the same cassette, right? So this looks like a cassette of multiple genes. So, and all of them will be, what do you say? All of them will be transcribed at the same time. So you will start here, RNA polymerase will come and sit here, and it will start the transcription, and it will go all the way till here. And when it will go all the way till here, there will be 3 mRNAs formed. One mRNA for gene 1, the other for gene 2, and the other for gene 3. Do you understand? For gene 1, gene 2, and gene 3. Is it clear, everyone? This is a polycistronic arrangement. Now here there is no split gene complexity. And why do you think, what is the difference? And can you think of one advantage that monocistronic system gives and one advantage that polycistronic system gives? Quickly. People. What is the benefit of having a promoter and a terminator for every single gene in a monocistronic arrangement?
Maybe because one type of RNA will be found.
Yeah, right. That's what is the outcome. Correct. But what is the advantage of having? Why is polysystronic system not present in us? Just have one promoter, one terminator, in between put 5-6 genes and just transcribe all in one go. So you understand that this kind of a system, polysystronic structural gene system, will save a lot of space, right? You don't have to put a promoter sequence, a terminator sequence after each and every gene. You don't have to make it a split gene arrangement, because see, at the end, when you are going to make a protein, the sequence of introns does not matter. Anyways, the transcription machinery will cut it out and remove it and join all the exons. All that a bacteria system already has. It does not care about introns. There is no introns. Everything is coding sequence. On top of it, it's not just one gene that you are transcribing. It could be multiple genes which you have fixed or you have set under the same cassette of a promoter and a terminator. So saves effort, saves space. And that's exactly why prokaryotes have polysystronic arrangement, because their genome is very, very small. They don't have non-coding sequences like human. I hope you know this, that 98%, approximately 98%, even slightly more than that, of our genome does not code for proteins. Do you know that? It's not a coding sequence. So 98% of our genome is not transcribed. So earlier we used to think that it was named as the junk DNA. So we have a lot of junk DNA, and our important proteins are very, very far away from each other on the chromosome, okay, on the sequence, not on the chromosome, sorry, because DNA folds and comes close. So on chromosome, they can be present very, very close, but on the long thread of DNA, genes are dispersed. And in between is the junk DNA that does not do anything. But now we have started figuring out that that's not true. All this DNA is not junk. Just like promoter and terminator, there are other regions called enhancer regions in the DNA that themselves do not get transcribed, but it affects the transcription of the genes. Okay. That is a different topic. I'll teach you sometime later, but just know that 98% of our genome is non-coding. It doesn't code for proteins. Okay, it codes for it's just other, it has other accessory functions, I will say. In bacteria, that's not the case. They have cut-to-cut genes put under these polysystronic cassettes, and a bacteria can make multiple genes expressed with just one promoter and one terminator as control. The benefit, the pros of this is saves space, and it's multiple genes can be transcribed at the same time. But the problem is regulation of each and every gene. Suppose you just want gene 1, but not gene 2 to be translated. Can you do this in a polysystronic system? No, you cannot. No one is speaking. Is the class very boring? No one is interested. Am I audible, people?
Yes.
Yeah. So can we, so suppose if you want gene 1 to be expressed and not gene 2, can you do that in a polysystronic structural gene? No, right? Because once it starts from the promoter, it will go all the way till terminator, and all these 3 will be transcribed. The regulation part can be done in a monosystronic system, because every cistron is monosystronic. It has only one gene, that also is interrupted with introns. So there is lots of regulation that you can do when one gene is expressed. Okay, and monocystronic system is only present in organisms whose genomes are very, very huge and big, and there is no problem of space. Okay, like eukaryotes. Prokaryotes have a problem of space. Mostly they have just one single DNA or one single chromosome, so they have to fit everything inside it. We have 23 chromosomes, so we have a lot of space. Okay, it's like 1 BHK versus a 23 BHK apartment. So imagine the space difference that we and bacteria have. Does that make sense, everyone? Monocystronic and polycystronic system?
Yes, sir.
Okay, very good. So what to now do? So let's talk about bacteria first. So similarly, we are going to study two different kinds. They're not very different. They're very, very similar, except for the part where this mRNA is processed. So first, let me tell you the process of transcription in prokaryotes. Now you know that in prokaryotes there is polycystronic. 1 second. Go up, go up. Yes. So in bacteria, what happens is there are 3 steps. Can you all come to open your NCERT? Let me pull out a figure. Open your NCERT and come to this process of transcription in bacteria. I'll just stop sharing for a moment. I'll try to pull out that image. Transcription in bacteria. You all see it? Also, please tell me what is the page number so that. Yes, I pulled it out. I hope you all can see my screen. Yes, this figure. Can you all see this figure in your note in your textbook?
Yes.
Yeah. What page number it is? It's figure 6.10.
109.
Sorry. 109. Yep. Right. Come to page 109 and look here. So in prokaryotes, there are 3 steps: initiation, elongation, and termination, simply. And the important thing that you have to know, so this is DNA. This yellow region is, they are highlighting as the promoter. So this is the promoter region where this RNA polymerase, so this big molecule, DNA dependent RNA polymerase, comes and sits. Sorry, I lost electricity, so bear with me. But I'm audible, right? Everyone?
Yes, sir.
Yeah. So RNA polymerase comes and sits at the promoter region, but how will RNA polymerase know where is the promoter region? And now I have to sit on it. So for this, there is this factor, a protein called sigma factor, which is also called as the initiation factor. Write down. Sigma factor is a protein which is called initiation factor. So this sigma protein binds to polymerase and activates polymerase, and now polymerase can come and sit on the promoter. Okay, and once it sits on the promoter, the transcription begins, and that's called the initiation step. So in the initiation steps, sigma factor comes and binds to RNA polymerase. Now this RNA polymerase is active and it can go and identify and bind to the promoter region. Okay, if the sigma factor does not bind to RNA polymerase, then RNA polymerase cannot identify and bind to the promoter region effectively. Is it clear, everyone?
It's
So it's something that brings the RNA pol to the promoter region. And then the next step is elongation, where parts of DNA keeps opening up, and then a small part of mRNA is synthesized, then it again closes, then again it moves forward, this opens, the RNA thread gets long, and this will close and it will move ahead. So it keeps sliding in between the two strands, and wherever it goes, the strand, let's say this is the strand, so strand opens, then closes, and then the mid part open and then closes. You understand? And the polymerase keeps sliding. Polymerase is a very big molecule. So basically, it keeps sliding, opens, make RNA, and keeps sliding. At this time, when it is doing elongation, the sigma factor is no longer required. So you can see that here, they have tried to show that the sigma factor is detached. Right? From RNA pol. Because sigma factor is only important for initiation. It is not required for elongation. It can keep going. Last thing is termination. When it reaches the termination site, which is this site here, let me make it in red. This, can you see this region? Where the terminator region. So when RNA pol reaches terminator region, again, it does not know when to stop. So another protein called Rho factor, which is also called as the termination factor or termination protein. This comes and binds to polymerase, and it's like tells the polymerase, okay, your job is done, get off the DNA now. And as a result, the RNA is released. So this is the RNA that was transcribed from this whole gene, from here till here. Does it make sense, everyone? So remember these factors, sigma and rho. Rho factor is for termination, sigma factor is for initiation. When it is in between elongating the mRNA, it does not need either sigma or rho. So polymerase is on its own. And this is DNA-dependent RNA polymerase. And the speeds at which polymerases work is very, very fast. It is approximately 2000 base pair per second, both for replication and transcription. You can imagine for transcription, it's still a little slower than replication, but 2000 base pairs per second is amazing speed, isn't it? It's like counting from 1 to 2000 in 1 second. And that's how fast molecules can work. Okay, so this is for DNA-dependent DNA polymerase that I'm telling you. DNA-dependent RNA polymerase is also very, very fast, somewhere around it. And how did we calculate it? Any idea? Can we count something 2000 times in 1 second? We cannot put a slow motion camera there inside the cell. So how do we know it's that fast? Anything that I tell you or the book tells you, you people are just conditioned to accept it as a fact. Remember, there's a difference between science and storybooks. Storybooks tell you stories, so you have to believe in it because that's how the story is narrated, right? You do not ask why there was a horse and not an elephant, because it doesn't make sense. The story is all about a horse, so it's a horse. So just keep your mind shut and listen to the story. Science is not a story. It's evidential. So you have to ask, how do we know it? How do we know the speed of DNA-dependent DNA polymerase that replicates the DNA? Any idea how can one know that? Because the people who figured it out were just like you, students of science. Now we call them scientists because they started thinking, solving problem one by one, and they became scientists. That's how we become scientists in today's world. You be curious about science, you get a formal education, you solve few problems in science, and you're a scientist. So if I ask you, so sadly for you, someone has already done it, solved it. But if this question came to your head hundreds of years ago, that how do you think, how fast is DNA polymerase? How will you do that experiment? It's simple mathematical calculation. Anyone? Any bells ringing in your head? Any neurons firing? Hiram, Mariam, Aisha? No one? Well, I know you people are just not trying. That's a bad thing. Bad thing is not that you are not giving the right answer. Bad thing is you are not even trying. It's simple. So what they did, they took bacteria. They first sequenced the bacteria. They got to know that bacteria has X number of base pairs. Then they cultured bacteria and calculated the time which is required to double its DNA and divide. So we know that E. coli divide every 25 minutes. So in that 25 minutes, it must have replicated its total DNA. And we already know how many base pairs are there and also have done other things. So when you get an approximate time and you know the number of base pairs, you can just divide the total number of base pairs by total time and you will get X number of bases per unit time. It could be minute, it could be per second. Do you understand, people? Yes, no, something will be great. Why is everyone so silent today? I'm suspecting some of you have already slept. Okay, Ali and Aisha are awake. I'm not sure if Parker and Mariam are awake or asleep. Anyways, so okay, they both are also awake, just not speaking. So that's how we get to know of the rate of these molecules or the work. So is transcription in prokaryotes clear to you?
Yes, sir.
It was very simple. Sigma factor binds to RNA pol. RNA pol comes and sits at promoter region. Then sigma factor goes away. RNA pol starts sliding and start elongating the mRNA, forming the RNA. And as it reaches the terminator region, a rho factor comes and binds. So rho factors are waiting around terminator region, comes and binds, take off polymerase. And as a result, the RNA is released. Okay, so we have a RNA from a DNA now. Transcription is done. In eukaryotes, it has two complexities in the process. So this was simple transcription in prokaryotes. And for this, I'm giving you a homework to read this paragraph from NCERT. And make notes. You understood the process? If you find any difficulty in making notes, just let me know. I'll dictate you notes in the next class if you cannot do it. Okay, everyone?
Yes, sir.
Yes, sir.
Now, since there is no split gene arrangement and no complexities of having very huge genomes, prokaryotes are simple. But when we go to eukaryotes, there are two complexities. One complexity is In a eukaryotic nucleus, scientists have figured out that there are more than one type of RNA polymerase. Okay. In prokaryotes, there is only one type of RNA polymerase, which is this one, which simply works. In eukaryotes, there are 3 RNA polymerases that you have to study. So 3 types of RNA polymerase are there. So which RNA polymerase transcribes what is the first complexity. And the second complexity is that because there is a split gene arrangement, when the RNA will be formed after transcription, it will have exons and introns. So you have to cut introns out and stitch exons together. So which is called post-processing of mRNA, which is not required in prokaryotes. So these are the 2 complexities. We will talk about both of these complexities in the next class. And with that, we will finish transcription and start the last thing, which is translation in the central dogma. Is that clear, everyone? Any doubts? Please, if you have any doubt, we have a couple of minutes left. So please bring up your doubts. I will need some water. Yeah, any doubts, people? Yes, no?
No, sir.
No doubt. Okay, so let's not start eukaryote today, because we'll take some time to finish. So let's stop here. Please go back and revise. There was minimal participation in today's class, which is not very good. So please speak up from next class onwards. Otherwise, I will not know what you understood and what you did not. And give answers, even if they are wrong. Do not worry. Please make this class a little more fun for me. Okay. Cool. I'll see you in the next class then.